Welcome to Aexon Lab's scientific meeting, where we explore the promise of comprehensive treatment for narcolepsy and related disorders. We are delighted by your interest in our innovative research aimed at targeting multiple mechanisms. Aexon Labs was established 2 years ago with a mission to discover breakthrough therapeutics that fully address the treatment of narcolepsy, Gilleneux's disease. Our goal extends beyond improving excessive daytime sleepiness and cataplexy. We strive to alleviate all symptoms of narcolepsy and significantly prevent or limit the apoptosis of orexin-producing cells. In this scientific meeting, we will share our significant achievements and highlight our recent patent applications, including one filed in December 2022 and 2 divisional applications submitted in December 2023. Notably, our molecules are not sulfonamide derivatives, unlike most currently developed OX2R agonist molecules, and we will explain the reasons behind this strategic choice. This presentation contains forward-looking statements and information that are intended for informational purposes only. Please refer to the detailed disclaimer for more information on the limitations and legal considerations. Aexon Labs is committed to developing innovative therapeutics designed to target multiple mechanisms offering precise and comprehensive therapeutic effects. Our focus is on managing and potentially reversing symptoms of narcolepsy and other neurodegenerative disorders using cutting-edge scientific techniques and rigorous pharmacological evaluations. Our current pipeline includes several promising candidates targeting both orexin receptors and cathepsins. These compounds show significant potential in preclinical studies for improving wakefulness, sleep quality, and reducing cataplexy attacks. We are committed to advancing these therapies through rigorous scientific research and development. Aexon Labs aims to build on this legacy by innovating towards an ideal treatment inspired by past research and discoveries on orexin neurons and their functions. Narcolepsy is a neurological disorder that involves much more than just excessive daytime sleepiness. Its pathophysiology is complex, and the term narcolepsies, used by Lermite in the early 20th century, more accurately reflects its various etiologies. Historically, Van Diemerbroek, a Dutch anatomist, described in 1664 that individuals with irresistible somnolence often experience sleep paralysis, during which they cannot move or speak. During these sleep attacks, they may see vivid and frightening hypnagogic dreams because they are not fully asleep. Jean-Baptiste Édouard Gilleneux, a pioneering French neurologist, coined the term narcolepsy in the late 19th century. He significantly advanced our understanding of this complex disease, which involves disturbances in REM sleep circuits and the loss of orexin-producing neurons in the lateral hypothalamus. Historical accounts from the 17th century to the early 20th century highlight the multifaceted pathophysiology of narcolepsy, emphasizing its chronic nature and the need for lifelong pharmacological management. Narcolepsy is a chronic neurological condition that necessitates lifelong pharmacological management. It predominantly affects young individuals and is characterized by the irreversible loss of orexin-producing neurons in the lateral hypothalamus. This neuronal destruction is central to the disease's pathogenesis and is not compensated by neuroregeneration. Narcolepsy is more than just uncontrollable sleepiness. It includes cataplexy, an abrupt loss of muscle tone triggered by strong emotions, and is an autoimmune and neuroinflammatory disorder where the orexin-producing cells are targeted and destroyed by the immune system. The pathophysiology of narcolepsy extends beyond a mere OX2R, orexin receptor 2 deficiency in the brain. A comprehensive understanding of its complex mechanisms is essential to develop effective treatments. The interplay between autoimmune processes, neuroinflammation, and sleep regulation circuits highlights the need for a multifaceted therapeutic approach to address the full spectrum of this debilitating disorder. I invite you to obtain the notebook on the illustrated history of narcolepsy to immerse yourself in the historical events that have shaped our current understanding. To address the therapeutic challenges of narcolepsy comprehensively, it is essential to adopt a multifaceted strategy that goes beyond current limitations. Our approach at Aexon Labs focuses on comprehensive symptom management by developing dual orexin receptor agonists targeting both OX1R and OX2R. This strategy aims to better mimic the natural orexin system, addressing not only wakefulness but also emotional disturbances associated with narcolepsy. Additionally, incorporating therapies that tackle multiple aspects of the disorder, including neuroinflammation and the preservation of orexin-producing neurons, is crucial. By overcoming the limitations of current treatments, such as the reliance on sulfonamide derivatives, we aim to provide more effective and holistic solutions for patients with narcolepsy. Explanation of receptor desensitization. Due to prolonged exposure to high-affinity agonists, normal receptor function. Receptors are proteins located on the cell membrane that bind to specific molecules, such as agonists. When an agonist binds to the receptor, it activates the receptor, leading to the coupling of the receptor to G proteins inside the cell. This coupling initiates downstream signaling pathways that produce a physiological response. Prolonged agonist exposure. Continuous exposure to high-affinity agonists leads to sustained receptor activation. Over time, the receptor becomes less responsive to the agonist. This is known as receptor desensitization. Reduced responsiveness. A desensitized receptor no longer efficiently couples to G proteins, inhibiting downstream signaling. As a result, the efficacy of the drug decreases over time, necessitating higher doses to achieve the same effect, which can lead to safety concerns. Moderate affinity, agonists, but targeting on OX2 or OX1R seems idealistic. By example, develop agonists with EC50 values between 10 nm and 300 nm to minimize receptor desensitization. Combination therapies. Use combination therapies to balance receptor activation and reduce the likelihood of tolerance. Non-sulfonamide compounds. Prioritize non-sulfonamide compounds to avoid known risks such as hypersensitivity and hepatotoxicity. Long-term studies. Conduct extensive long-term studies to ensure safety and efficacy over prolonged use. To address the therapeutic challenges of narcolepsy comprehensively, it is essential to adopt a multifaceted strategy that goes beyond current limitations. Our approach at Aexon Labs focuses on comprehensive symptom management by developing dual orexin receptor agonists, targeting both OX1R and OX2R. This strategy aims to better mimic the natural orexin system, addressing not only wakefulness but also emotional disturbances associated with narcolepsy. Additionally, incorporating therapies that tackle multiple aspects of the disorder, including neuroinflammation and the preservation of orexin-producing neurons, is crucial. By overcoming the limitations of current treatments, such as the reliance on sulfonamide derivatives, we aim to provide more effective and holistic solutions for patients with narcolepsy. Since the first application claiming orexin receptor agonists, the importance of the sulfonamide group for activity at orexin receptors has been clearly indicated. Many major companies have focused specifically on selectively targeting OX2R. However, this approach may not be the best long-term option. Sulfonamide-based molecules targeting OX2R, such as those developed by Takeda, have encountered significant safety issues. TAEX-994, for instance, was associated with liver problems, and switching to a similar compound, TAC-861, has only resulted in different side effects like frequent urination, despite the addition of a fluorine substitution. Focusing solely on OX2R may disrupt the balance between OX1 and OX2 receptors, potentially leading to a loss of effect related to food intake, as reported by Evans in 2022 with TAC-925. This compound does not cross the blood-brain barrier, BBB, requires continuous injection throughout the day, and shows low efficacy in maintaining wakefulness during meals. At Aexon Labs, We avoid these pitfalls by not concentrating our research on sulfonamide-type compounds, aiming instead for a more balanced and safer therapeutic profile. While sulfonamide-type compounds may be better at selectively targeting AEX, these chemical structures are notorious for causing hypersensitivity reactions, including severe conditions such as Stevens-Johnson syndrome. Patent applications often downplay these risks or fail to provide comprehensive data on mitigating these adverse effects in clinical use, which is a significant drawback when evaluating the long-term viability of these compounds for chronic conditions. The FDA has previously noted that long-term use of sulfonamide-based drugs can lead to accumulation and potential toxicity, particularly affecting the liver and kidneys. Patents should provide detailed studies on the metabolic pathways and excretory mechanisms of these compounds to ensure they do not pose significant risks of toxicity over prolonged periods. While the ability to cross the BBB is necessary for CNS drugs, sulfonamides that do cross the BBB must be evaluated for their potential neurotoxic effects. Chronic exposure to sulfonamide derivatives in the CNS could lead to neuroinflammation and other adverse neurological outcomes. Patents need to include thorough investigations into the long-term effects on CNS tissue to address these concerns adequately. Aexon Labs is committed to neuroscientific innovation and has developed first-in-class non-sulfonamide dual orexin receptor agonists, exploring the potential of targeting cathepsins to prevent orexin cell apoptosis. In narcolepsy, the loss of orexin-producing neurons is a critical factor contributing to the disorder's symptoms, including excessive daytime sleepiness and cataplexy. HLA class II histocompatibility antigens, such as DQ-beta-1, play a significant role in presenting antigens to CD4 T cells, which can lead to the autoimmune destruction of orexin neurons. This autoimmune response is a key pathological mechanism in narcolepsy. By inhibiting cathepsins, particularly CTSH, CTSB, and CTSD, we aim to modulate the immune response within the central nervous system. Cathepsins are proteolytic enzymes involved in antigen processing and presentation. Inhibition of these enzymes may reduce the activation of CD4 T cells and subsequently decrease the immune-mediated damage to orexin neurons. This approach not only addresses the autoimmune component of narcolepsy, but also helps to mitigate the detrimental effects of neuroinflammation, which is a common feature in various neurological disorders. Aexon Labs is committed to neuroscientific innovation and has developed first-in-class non-sulfonamide dual orexin receptor agonists integrated with cathepsin inhibitors. These compounds target multiple mechanisms, including OX1R and OX2R, as well as cathepsin inhibition. This multi-target approach aims to address the complex pathophysiology of narcolepsy and related disorders by promoting wakefulness, improving daytime alertness, and potentially offering neuroprotective effects. Our findings, supported by applications filed since 2022, EP2230775, and a patent cooperation treaty, PCT 2023-088020, relate to the use of dihydroquinazolone, DHQ, and dihydrobenzothiazine, DHB derivatives for the prevention or treatment of neurological diseases, psychiatric disorders, and sleep disorders. These conditions are particularly relevant when lateral hypothalamic neurotransmission of orexin is compromised and upon engagement of OX1R and/or OX2R receptors. These inventions highlight the importance of our innovative non-sulfonamide heterocyclic compounds in the treatment of narcolepsy. The main results on in vitro and preliminary studies have been presented at the ASCP 2024 annual meeting. You can download this poster by clicking on the link below. With this poster, we present our groundbreaking research on non-sulfonamide dual orexin receptor agonists and cathepsin inhibitors from Aexon Labs. As you know, current effective treatments target several systems, including catecholamines. For example, mazindal, a pan-monoamine reuptake inhibitor, and partial OX2R agonist, has been shown to be significantly effective in reducing excessive daytime sleepiness and cataplexy. FDA-approved treatments like modafinil, solrium fatal, and amphetamine salts also inhibit catecholamines. Other treatments, such as pitolisant, act on histamine 3 and sigma-1 receptors, and have also shown efficacy in treating narcolepsy symptoms. The sigma-1 receptor is also targeted by Aexon Labs to help treat symptoms of narcolepsy with cataplexy. Compounds such as AEX1, AEX24, and AEX5 are already focused on this therapeutic strategy. Finally, we believe that future innovative treatments for the tetrad symptomatology of narcolepsy should be multi-target, as is often the case in psychiatric medications such as fluoxetine, aripiprazole, quetiapine, and others, which have demonstrated efficacy on various therapeutic targets over time. In this poster, we presented in vitro studies conducted by Eurofins Discovery, highlighting the potency of various Aexon Labs compounds, particularly AEX1 and AEX2, in activating orexin receptors and inhibiting cathepsin. The radar plot illustrates the pharmacological profiles of various AEX compounds, with AEX1 showing high potency across multiple targets. These compounds also inhibit catecholamine transporters such as DAT and NET, corroborating the wake-promoting efficacy of AEX1 and AEX2. Additionally, AEX1, AEX2, and AEX4 act on cathepsin H, CTSH, involved in the pathophysiology of narcolepsy, by restoring orexin receptor function, preventing apoptosis of orexin-producing neurons, and balancing neurotransmitter levels via cathepsin H inhibition. After these promising in vitro results, we presented our preclinical development plan, led by Professor Christelle Perron, who participated in the discovery of orexin neurons in 1998 and is a research director at the Lyon Neurosciences Research Center, previously directed by Professor Michel Jouvet. Jouvet discovered in 1957 that REM sleep was associated with unique physiological and behavioral characteristics, including rapid eye movements, muscle paralysis, and vivid dreams such as hypnagogic dreams reported in narcolepsy. Orexin knockout mice will be used to mimic human narcolepsy, testing wakefulness, sleep quality, and cataplexy suppression. Preliminary results are expected to highlight that AEX compounds effectively manage narcolepsy symptoms, potentially outperforming selective AEX2R agonists. Previous competitive studies with experimental sulfonamide compounds, sometimes injectable, have shown that selective AEX2R agonists fail to maintain their effectiveness after food intake due to increased orexin A levels, which do not integrate well with the OX2R agonist treatment regimen. Therefore, we believe that targeting both AEX2R and OX1R offers a long-term advantage. This strategy is supported by Takeshi Sakurai, co-discoverer of the orexin system, who emphasized in a 2023 publication in Peptides that AEX1R plays an additional role in the regulation of wakefulness. Rescuing the function of both receptors would be ideal. Our current pipeline includes several promising candidates targeting both orexin receptors and cathepsins. These compounds show significant potential in preclinical studies for improving wakefulness, sleep quality, and reducing cataplexy attacks. We are committed to advancing these therapies through rigorous scientific research and development. For example, AEX2, AEX3, and AEX41 are designated for narcolepsy, showing great promise in our studies. Additionally, other compounds in our pipeline, such as AEX5, AEX19, and AEX23, are focused on neurodegenerative diseases like Parkinson's disease. Notably, AEX19 could potentially be effective in treating tauopathies, such as Alzheimer's disease, through its action on the sigma-1 receptor. Our divisional application on therapeutic indications highlights the breadth of our research. The first wave of studies, led by Christelle Perron, will focus on the evaluation of compounds AEX2 and AEX41. These studies aim to further validate the efficacy and safety of our compounds in managing narcolepsy symptoms and improving overall neurological health. Our research highlights the importance of targeting both orexin receptors and cathepsins to address the full spectrum of narcolepsy symptoms. We believe that our multi-target approach will provide more effective and safer treatment options for patients with narcolepsy. Aexon Labs is committed to developing dual orexin receptor agonists targeting both AEX1R and AEX2R, offering a multi-target approach to address wakefulness and emotional disturbances. Our research integrates cathepsin inhibitors to reduce neuroinflammation and protect orexin-producing neurons, exploring immunomodulatory treatments to prevent further neuronal damage. We prioritize moderate affinity agonists with EC50 values between 10 nm and 300 nm to minimize receptor desensitization, employing combination therapies for balanced receptor activation and reduced tolerance. Aexon Labs focuses on non-sulfonamide compounds to avoid risks such as hypersensitivity and hepatotoxicity, conducting extensive long-term studies to ensure safety and efficacy. Our patient-centric approaches tailor treatments based on individual profiles, implementing regular monitoring to adjust treatments and manage side effects effectively. We develop optimized formulations, ensuring optimal bioavailability and CNS penetration, utilizing advanced drug delivery technologies for precise targeting and controlled release. Aexon Labs adopts a holistic and integrative scientific approach to find the ideal treatment for narcolepsy. Our commitment to addressing both the neurological and autoimmune components of the disease while prioritizing safety and efficacy reflects our dedication to improving patient outcomes